Zat-zat yang masuk dan keluar melalui membran sel. Membran sel menjadi pembatas antara isi dan lingkungan yang ada di luar. Membran sel bersifat semi-permeable atau juga disebut sebagai selective permeable. Yang artinya membran sel melakukan seleksi terhadap zat yang masuk dan zat yang keluar. Struktur lipid membran sel terdiri dari fosfolipid yang membentuk bilayer. Struktur bilayer ini ada karena adanya ekor dari dua fosfolipid yang saling berhadapan. Ekor-ekor ini bersifat hidrofobik, yaitu sifat fisik dari suatu molekul yang tampaknya ditolak dari massa air. Berarti membran sel ini tidak menerima selain nonpolar seperti lemak dan glukosa. Di antara dua lipid bilayer ini disematkan protein-protein integral atau protein yang menempel pada lipid bilayer. Selain protein, pada beberapa membran sel juga mengandung kolesterol. Membran plasma adalah penjaga gerbang sel yang mengizinkan zat tertentu masuk dan keluar sel pada jumlah tertentu dan waktu tertentu. Berbagai macam transport terjadi di membran sel. Secara garis besar, terdapat transport aktif dan transport pasif. Sekarang kita akan membahas
with no energy required, while active transport needs energy for it to occur.
Terdapat transport aktif dan transport pasif. Sekarang kita akan membahas transport pasif. Di antaranya ada difusi dan difusi terfasilitasi. Yang pertama ada difusi, yaitu proses perpindahan zat melalui membran sel dari area berkonsentrasi tinggi ke area berkonsentrasi rendah tanpa memerlukan ATP. Partikel kecil dan tidak bermuatan seperti oksigen dan karbon dioksida dapat berdifusi melalui membran plasma dengan bergerak melalui fosfolipid pada bilayer. Yang kedua, difusi terfasilitasi, yaitu molekul yang lebih besar seperti molekul polar membutuhkan protein karier khusus yang akan mengikat zat tersebut pada salah satu sisi membran, yang kemudian protein tersebut akan mencetuskan serangkaian proses konfirmasi diterimanya molekul, sehingga dapat melewati membran lalu masuk ke sel sisi yang lainnya, contohnya seperti glukosa.
Across a semi-permeable membrane. This membrane is semi-permeable because water molecules can pass through it. But solute molecules cannot because they are too large. The beaker on the screen shows a semi-permeable membrane separating a solution with a low solute concentration on the left from a solution with a higher solute concentration on the right. There is an overall passive movement of water molecules from the solution with a low solute concentration to the solution with a high solute concentration. This is because the water molecules move to even out any difference in concentration, just like in ordinary diffusion. This net movement of water molecules will cause the water level to rise on the right and fall on the left. Hydrostatic pressure is the pressure exerted by a fluid due to the force of gravity acting upon it. As the column of water on the right gets higher, the hydrostatic pressure it exerts increases. The hydrostatic pressure acts to push the water molecules back across the membrane, opposing the movement of water by osmosis. At some point, this pressure is high enough to stop the net movement of water through the membrane by osmosis. This pressure is known as the osmotic pressure. The osmotic pressure is determined by the concentration of non-diffusible solutes in the solutions. If there is a small difference in solute concentration between the two solutions, there is a low osmotic pressure. In this case, a small amount of water crosses the membrane by osmosis, raising the level of the solution on the right slightly. As a result, the hydrostatic pressure increases slightly, which is enough to prevent any further net movement of water. However, if there is a large concentration difference between the two solutions, there is a much higher osmotic pressure. A large amount of water can cross the membrane, raising the level of the solution on the right considerably, before the hydrostatic pressure will be enough to prevent further net movement of water.
Needs energy for it to occur.
Detail. So, there are two main forms of endocytosis, pinocytosis and phagocytosis. First, let's talk about pinocytosis. Pinocytosis means drinking by the cell. It occurs to take in macromolecules. The cell expresses receptors for such molecules on the membrane. Such receptors are generally concentrated in a small pit on the membrane. It is called a coated pit. Beneath this pit, on the inner side of the cell, there is a latticework of fibrillar proteins called clathrin, and other proteins, like contractile filaments, actin and myosin. All these proteins drive the movement of the cell membrane during endocytosis. What happens is, when the particle attaches to the receptor on the outer side of the cell, these proteins pull the membrane inside the cell. This deepens the pit. Then they close the mouth of this pit, forming a vesicle inside the cell. Then these proteins dissociate from the vesicle and go back to the membrane for next round of endocytosis. The entire process requires energy from ATP. The vesicle formed by pinocytosis is called a pinocytotic vesicle. Such vesicles are very small. Pinocytosis occurs in almost all the cells in the body. So this was about pinocytosis. Now let's talk about phagocytosis. It means eating by the cell. It occurs for the intake of large particles, such as bacteria or whole cells, etc. And only some cells are capable of doing phagocytosis, like tissue macrophages and some white blood cells. The process of phagocytosis is similar to pinocytosis. First, some ligand on the particle to be engulfed binds with the receptor on the cell. This initiates the process of phagocytosis. In this, the membrane around the points of attachment evaginates outwards to surround the entire particle. At the tip, the membrane merge, creating a vesicle inside the cell. It's called a phagocytic vesicle. So this is how phagocytosis happens. Now, the cell has organelles called lysosomes, containing various hydrolase enzymes. The vesicle formed by pinocytosis or phagocytosis merges with this lysosome. This empties the enzymes and lysosomes in the vesicle. Now it's called digestive vesicle. And as per the name, it starts digesting the proteins, carbohydrates, lipids, and other things in the vesicle. Small molecules like amino acids, glucose, phosphate, etc. formed during this digestion cross the membrane and enter the cytoplasm. Indigestible things are left inside the vesicle. They are excreted out of the cell by exocytosis. So this was all about ingestion by the cell. Now let's have a quick summary.
The opposite of endocytosis is exocytosis. Exocytosis is when something needs to exit the cell. The cell can remove large molecules or wastes this way by fusing the membrane-bound vesicles containing them with the cell membrane, forcing them out of the cell. A good way to remember that exocytosis is a way for things to leave the cell is that it shares the first two letters with exit. You can also remember that endocytosis is a way for things to move into the cell because it shares the first two letters with enter. Sometimes the cell uses special protein pumps to move small molecules or ions against the concentration gradient into or out of the cell. An example of this is the sodium-potassium pump. In this process, the pump uses energy in the form of ATP molecules to move sodium ions out of the cell, and then move potassium ions into the cell. Protein pumps used in active transport require energy because the molecules or ions are moving from an area of low concentration to high concentration.